Hello, everyone. This is going to be our first Zoom recording for the semester. Since I have uploaded the PDF file on macromolecules, I'm not going to go in detail, so I'll try to finish the slide within 10 minutes or less. So macromolecules. Uh, Macromolecules are the building blocks of all living organisms. Uh, yeah, everything in our surrounding, every living thing is made of macromolecules. And these macromolecules include carbohydrates, proteins, and lipids. And carbohydrates, they have monomers and polymers, and probably you are familiar with the word monomer. When we say monomer, it means it is the simplest form of a certain kind of macromolecule. So for example, for carbohydrates, the monomers are monosaccharides, and some examples of monosaccharides include glucose, galactose, ribose, all the sugars, the simplest sugars we know, they are the monomers. When these monomers are joined together through a hydrolysis reaction, we get disaccharides. One simple example for a disaccharide is sucrose or a table sugar. The other one is lactose. This is a disaccharide we find in milk. And also we have galactose. Oh, I'm sorry, galactose is actually simple sugar. So we have sucrose, lactose, and fructose. Fructose is the one we find in fruits. Yep, so these are the saccharides. Another name for disaccharides could be oligosaccharide, because oligosaccharide includes any kind of carbohydrate that has two sugars or more. So sucrose could be a disaccharide and at the same time oligosaccharide. The only difference for oligosaccharide is it could contain more than two sugar molecules. On the other hand, polysaccharides are the polymers of carbohydrates or the polymers of monomers or single sugar molecules. One typical example for a polysaccharide is starch, and another one is glycogen. Glycogen is found in animals and starch is found in plants. Carbohydrates have their own functions. The first function is storage. And as I told you, the form of storage in plants, the form of storage for plants use starch to store carbohydrate and animals use glycogen to store sugar molecules. And the other function of carbohydrates is for energy storage, obviously, and most of the energy we use in our body and the energy that plants use comes from glucose. And if you wanted to know, let's say if you are interested to know if a certain sample contains carbohydrate or not, all you can do is you can run a couple of tests and you can tell whether that sample contains starch or reducing sugars or any other carbohydrate molecule. So for starch, you can run iodine test, and on Connect, I think there is one simulation that shows you how to run an iodine test. It's just a liquid. Iodine is a liquid. All you have to do is take a sample of starch or an unknown solution, and then you add a drop of iodine on it. And if there is a change in the color of the solution after you added iodine, not just the color of the iodine, but the whole solution, then you can tell that unknown solution has a starch. Usually, the darker the result, the more carbohydrate content. Because iodine is yellow, so if the color is changed into darker colors, let's say like dark black or purplish color, that means that molecule has starch. Another test that you can do to know if a certain molecule has reducing sugars is Benedict's test. When we say reducing sugars, actually, we are talking about disaccharides and oligosaccharides. So when we say reducing sugar, as the name implies, these are sugars that help in the reduction of other molecules they interact with. You don't need to know, but when we say reducing sugars, just it is implying a redox reaction. It is these are sugars that help in the reduction of other molecules that they interact with. The same thing for Benedict's test. It is a light blue solution. So what you do is you take a sample of carbohydrates and you add this light blue solution on it. And if it changes color into a darker solution, that means the sample has some kind of reducing sugars. And one example for reducing sugar is maltose, and another one is glucose. The second kinds of macromolecules are proteins, and the monomers of proteins are called amino acids. And when these amino acids are joined together, they form dipeptides. And when these dipeptides join together, they form polypeptides. Polypeptide is another name for a protein. You know, lay people call it protein, and in biology we call it polypeptides. Proteins are one of the macromolecules that have really numerous functions, and the first function of proteins is catalysis. And when you think of catalysis, always enzymes come up because enzymes are called biological catalysts. They are really important in speeding up a biological reaction. For example, if we take amylase, which is found in our intestine, small intestine, if I'm not mistaken, as well as in our mouth, it breaks down starch into glucose. So proteins are very important or very vital in this case. In terms of structure, one typical example is collagen. Collagen is the main component of our bones. So here too, proteins play a very vital role. Another one is transport. And as you all know, hemoglobin is one of the prime examples about the role of proteins in transport. And there are also other transport proteins like dynein and kinesin. Another function of protein is signaling. When you think of signaling, always glycoproteins come up. And as you can see, it has glyco and has protein in its name. These glycoproteins are found on the surface of cell membrane, or we call them surface proteins, cell surface proteins. And they are really important in receptor signaling. So, like the Benedict test and iodine test, proteins have their own taste. If you wanted to know, let's say, if a certain solution or a certain sample has a protein, you can run a burette test. All you do is you take a sample and you add a drop of burette reagent. And then burette reagent is usually light blue in color. So after the addition of the burette reagent, if the color of the solution is changed into darker colors, let's say purple, pink, then that means that solution has a protein. I'm sure there's an example or a simulation in Connect, so take a look at it. The last group of macromolecule we need to see for today are the lipids. Lipids are long hydrocarbon molecules. When you say hydrocarbon, it means they have a lot of carbon and hydrogens, and which makes them non-polar molecules. Because as you know, the polarity difference between hydrogen and carbon is almost 0.4 or less. So lipids tend to be they tend to be non-polar. The monomers of lipids Typical examples include glycerol and fatty acid. Glycerol is, in a sense, it is an alcohol, as you can see, it ends with oil. But since it has 3 carbons and it tends to be non-polar compared to other molecules, it's considered a monomer of lipid. Polymers of lipids include fats and oils. Don't confuse this one. Lipids are the larger category, and within it we have fats and oils. Fats are solids at room temperature, and we usually find fats from animals. Oils, we find them in plants, and they are always liquids at room temperature. As I have said earlier, since lipids have a lot of hydrocarbon molecules, they easily dissolve in non-polar solvents than polar. Non-polar solvents, in a sense, like in oil, fat, soap, things like that, instead of water or alcohol. When you think of lipids, there are certain topics that come up associated with it. The first one is the degree of saturation of the molecule. When we say a certain oil is saturated, we are talking, when we say a certain lipid is saturated, we are talking about the presence of carbon-carbon single bonds. So if a certain lipid is, or if a certain fat is saturated, we are saying there are only single bonds in there, single bonds of carbon-carbon single bonds. And when we say unsaturated, at least they do have one double bond. That means a carbon-carbon double bond. And because of that carbon-carbon double bond, there will be a kink in the molecule, which makes it less stable and, in a sense, less saturated. Another topic that comes associated with lipids is emulsification. Emulsification is, actually, it is easier to think of it as washing something with soap, but that's what it actually is. So in emulsification, there should be an emulsifier. An emulsifier is a molecule that has dual ends, that are polar and non-polar ends, and due to the presence of these polar and non-polar ends, these emulsifiers are good in breaking down large lipids into smaller fragments. And actually, even if you have a polar solvent along with the emulsifier, it's actually easier to break it down. Function. One of the major function of lipids is structure, and one typical example is the phospholipid bilayer. And as you probably know, cells have cell membrane, and that membrane is a double membrane system. And this double membrane is because of the phospholipid bilayer. And the other function is storage. A typical example is triglycerides. These are storage molecules for lipids. And actually, in some cases, glycogen could be converted into lipid, and that happens usually in association with triglycerides. And if you want to know if a certain solution, unknown solution, has fat or oil or in general lipid, you can run a paper test. In a paper test, it is exactly how it is read. It's a paper test. You take a piece of paper, and then you take a sample of unknown solution. You put it on. You put a drop of this unknown solution on the paper, and you let it sit for 15 minutes. So if it is a lipid, it doesn't evaporate. That means it is going to stay there, and you are going to have a large brown spot. It's oily and brown, you will see that. But if it was, let's say, water or alcohol, or something that's polar, it's going to evaporate, and after 15 minutes, you are not going to see anything. This is all I have regarding macromolecules. Yep. Thank you.